Here are some videos that you might find useful as you're reviewing this chapter. I'm just going to play them one right after another. So here you go.
In this diagram, we are looking at a simplified schematic of the types of reactions that occur within the chloroplast of a photosynthetic cell. The inputs shown here, water, carbon dioxide, and solar energy, or sunlight, are all coming from outside of the cell. We are going to focus on the light reactions, which are the first step of the photosynthetic process in most plants. As we proceed through these diagrams, notice how these reactions have inputs of solar energy and water. In order to understand the light reactions, it is often easiest to focus on the flow of electrons, shown here as blue circles. Let's start by exploring what is happening in Photosystem 2. Photosystems are collections of pigments that concentrate the incoming solar energy on a reaction center. This energy causes the release of high-energy electrons, which in turn are collected by an electron acceptor. The high-energy electrons, which contain the energy originally found in the incoming photons, are then passed to the electron transport chain, or ETC. The ETC harvests the energy from these electrons and use it to charge adenosine diphosphate, or ADP molecules, forming adenosine triphosphate, or ATP. Remember that this is a non-cyclic reaction, so these electrons are not returned to Photosystem 2. Instead, they are handed off to Photosystem 1. At the same time as these reactions are occurring in Photosystem 2, sunlight is also striking Photosystem 1, causing a similar release of high-energy electrons. However, unlike the electrons from Photosystem 2, which were used to generate ATP, these electrons are captured to reduce NADP+, forming NADPH. So, as you can see, this set of reactions is not cyclic, meaning that the electrons are not returned to where they started from. The electrons leaving Photosystem 2 are used as replacement electrons for Photosystem 1, while the electrons from Photosystem 1 are used to form NADPH. However, there is one small problem. Over time, Photosystem 2 would lose all of its electrons and disintegrate. But this does not happen due to the fact that some of the solar energy in Photosystem 2 is used to split molecules of water, releasing electrons. This is called photolysis. The electrons being generated during photolysis replenish the electrons being lost in Photosystem 2. Notice how some oxygen is also produced. This oxygen exits the cell as oxygen gas. Finally, we can take a look at what happens to the ATP and NADPH produced during the light reactions. Remember that this is only the first stage of photosynthesis, and that the overall goal was to capture the energy of the sun and produce carbohydrates. In this first set of reactions, we have captured solar energy and stored it temporarily as ATP and NADPH. Both of these molecules will now be used in the Calvin reactions in the formation of carbohydrates, or CH2O. Once used, they are recycled back to the light-dependent reactions as ADP and NADP+, respectively.
In most photosynthetic organisms, thylakoids contain pairs of photosystems, called Photosystem 1 and Photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH.
When glucose is oxidized during glycolysis and the citric acid cycle, NAD+ and FAD are reduced to NADH and FADH2, and hydrogen ions, H+, are released. In the mitochondria, the electrons from NADH are transferred to the electron transport proteins, and the hydrogen ions are transferred across the membrane. As the electrons move from carrier to carrier down the electron transport chain, more hydrogen ions are carried across the membrane. The terminal electron acceptor for this process is oxygen. When oxygen is reduced, water is produced. The transfer of hydrogen ions generates a potential energy gradient across the membrane of the mitochondrion. Because membranes are impermeable to ions, the hydrogen ions that re-enter the matrix must pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate by the ATP synthase complex.
Photosynthesis is a two-step process. The first set of reactions, called the light reactions, transfer the kinetic energy of sunlight into the molecules ATP and NADPH. In the second set of reactions, we are going to use this energy and carbon dioxide gas from the atmosphere to produce carbohydrates. As you proceed through the stages of the Calvin cycle, do not lose sight of the fact that the overall goal is to produce carbohydrates. The Calvin cycle begins with an input of 3 molecules of carbon dioxide, or CO2. Carbon dioxide is a gas, so to facilitate the following chemical reactions, it is combined with an organic molecule. That molecule is called RUBP, and the process is called carbon fixation. Carbon fixation produces a 6-carbon molecule, which shortly thereafter splits to form a pair of 3 carbon molecules called 3PG. The next stage of the Calvin cycle uses the energy products made during the light reactions to reduce the 3PG molecules. This stage requires an input of energy, which is supplied by ATP. Remember that reduction involves an addition of electrons, which in the Calvin cycle is supplied by NADPH. The addition of the electrons from NADPH reduces 3PG to form G3P. The ADP and NADP plus from this stage return to the light reactions to be recharged. G3P is an important intermediate of the Calvin cycle, since it is the precursor for the formation of many organic molecules, including carbohydrates such as glucose. At this stage, the plant has successfully harvested the energy from the sun and used it to reduce carbon dioxide gas to form carbohydrates. However, this process would be relatively inefficient if it were not possible to recycle some of the intermediate molecules, specifically RUBP. The last step of the Calvin cycle acts as its recycling phase. Some of the G3P produced earlier is modified using ATP, which originated in the light reactions. The RUBP produced by this stage may be used to fix more CO2, thus continuing the cycle.